Okay, so to begin, our intended target is colorectal cancer, which is the second cause of cancer deaths in the US currently, with a current case rate of 36.8 people per 100,000 cases as of 2017. The country with the worst rate is currently Hungary, well, as of 2018, with an age standardized rate of 51.2 per 100,000 people. But both of these rates can be lowered by screening for early detection. But not very many people get screened as often as they should. And some factors as to why that is include scheduling, difficulty with scheduling. People can be discouraged by cost or lack of insurance, lack of education regarding the subject, being afraid and or embarrassed by the current usual test procedures, including fecal occult blood testing, flexible sigmoidoscopy, colonoscopy, or barium enema. A lot of people are discouraged by the bowel prep that's involved with some of these procedures. Some people don't want to get screened or tested because they're not currently experiencing symptoms. And a lot of people avoid it out of fear of getting the bad news of having cancer. As a local application to this, Native Americans have the lowest of US colorectal screening rates, with 48.7% of 50 to 75-year-olds having gotten tested in 2015, compared to 65.4% of whites. And the reason for their low screening rates can be attributed to some of the same reasons from above. Currently, University of Arizona was given $3 million to combat this, but we hope to provide a convenient, lower cost, at-home testing method, which should overcome some of these inadequate screening factors.
And then, so just to kind of reiterate on the importance of this. So as she mentioned, colorectal cancer is the second most common type of cancer, but only about 58.2% of Americans aged 50 to 75 actually get screened for colorectal cancer. And the 5-year survival rate for colon cancer is listed below. So you kind of see how much it drops as the cancer spreads. And the kind of takeaway from that is that early detection saves lives. So it's important to, get tested.
All right, so there are some important stakeholders who are invested in this. For example, like Christina mentioned about how Native Americans have low testing rates. The American Indian Cancer Foundation is interested in raising more awareness about this. As well, the University of Arizona, their cancer center has the Cancer Moonshot Initiative to help encourage Native Americans increase screening. As well, at ASU in the biodesign, the Department of Personalized Diagnostics, they, I think, could be interested in this in our project. Specifically, these professors, they're all interested in biomarkers and antigens, and I think could help us in the future.
So for current methods of detection, so the important thing to notice is that many of the people that go to get tested for colon cancer are already in their later years of life. And the only tests that we have right now are like things like colonoscopy, we have schistometoscopy, we have CT colonography and stool DNA tests. And they often require you going to your doctor or your physician to get these tests. But the opportunity cost for these is that it's often cost effective. And it's only that people come for like late detection when the cancer is most visible. So if we can solve for these factors like cost or even like early detection, we have a large gain that we can make from this project.
So in terms of what we actually hope to accomplish with this in order to develop a colorectal cancer test is we would want to modify E. coli to detect high levels of CA19-9, which is a bioindicator for colorectal cancers, as well as some other cancers, and then express a fluorescent protein upon detection. So we would modify the quorum sensing system within E. coli to detect CA19-9, and then add a fluorescent protein gene with expression dependent on signaling from this system. And then we would want to ensure, obviously, the biosafety of the modified E. coli for use within people as well.
And that brings us to how we're going to actually plan out this experiment, which we've broken down into 4 major steps. First, we plan on using a heat shock technique to introduce the modified plasmon into the E. coli. Then we plan on setting up varying well plates with varying different sets of concentration of CA19-9. We then plan on taking 2 separate tests, first a visual indicator, then a light spectroscopy to ensure that the concentrations of GFP are prominent enough to be detected in fecal matter.
Okay, so for the cost of the supplies, of course, we're going to need E. coli, and online, I found that it will cost around $300 for each strain. We will need plasmids, and they cost around $75. And also ASU has the DN-ASU plasmid repository, which is local, and we could utilize their supplies. We're going to need the green fluorescent protein, which costs as well around $300. The antigen CA19-9 costs around $234 for 25 units. And we hope that if we get a lab on campus, that we could maybe utilize their wells for cells, and as well their spectrophometer for analyzing the concentration, and as well utilizing black light to actually see the fluorescence and see if it's working. As well, we wanted to discuss the ethics about in the future if this were to be utilized for humans. We hoped it would be used as a suppository, and as well there are risks with taking suppositories that could affect the bowel movements and system. As well, taking E. coli has certain side effects. You could have allergic reaction, it can affect the stomach and cause serious infection. Again, if way down in the future, if this goes into clinical trials, there would be ethical concerns with that and getting FDA approved. As well, with the fluorescent protein, this could interfere with the body and affect T cell activation.
So then that kind of leads to future research and end goals. So, this is more long-term and outside of our scope, but eventually, this project would hopefully work through animal models to ensure safety and efficacy. And then for the human use, we would administer the bacteria via suppositories. And this method would have a more broad detection than just colon cancer. But our hope is that it can primarily detect colon cancer. And then the short goal for this would be in vitro cell culture, and then with a long-term goal of cost-effective sampling to measure, potential colon cancer.
Curtis was talking about CA19-9 as an antigen is reflected of more than just colon cancer. As previously mentioned, it can be related to many different cancers, while also other gastrointestinal diseases that aren't cancer related. But we feel like this still folds into the scope of our project and still works as an early onset detection system.
Oh yeah, and then so some other possible alternatives that we might look at would be using a CA19-9 receptor for signaling, or potentially looking at another antigen as a biomarker, such as the carcinoembryonic antigen, as a potential indicator for cancers like colon cancer.
That's it for us.
Any questions? Or are we going to wait till after everyone's
Nope,
presented?
that was perfect timing. Thank you. The professors and I guess students, if you have questions also, have about 10 minutes until 5:25.
Okay. So what can you tell me about the CA19-9 antigen? I see that it's a sugar that glycosylates cell surface proteins and it's free floating. So there is a receptor for it already. There's already a signaling system known. That part I wasn't clear with.
Yeah, so this is one of the major things that we'd hope to overcome during the next phase of research. While we aren't certain that there is a receptor built in place, what we do know is that it's an antigen that's overproduced when in relation to cancers like colon cancer.
So Yeah, I mean, I think that was my question as well, was when, so you have E. coli, right? And then you have GFP, and somehow you're going to express GFP when CA 19-9 is present, right? In fecal matter or wherever it is that you're doing the assay, right? So how that's detected by E. coli is I think the big question, right? Like how does E. coli sense that and then turn that in transduce that into some sort of signal that basically then causes GFP expression? Is there a receptor out there for CA 19-9?
We aren't certain about the actual specifics about a receptor. We weren't able to figure that out because it's just a bit beyond our understanding of science or microbiology at this point. But we do know that the method we anticipate modifying is by taking the quorum sensing capabilities of E. coli and modifying the encoder receptors to be to copy whatever pre-existing ones could exist. And one of the things I think is really important is that we aren't certain that CA 19-9 would be the best, but we figured that this is our best shot within the limited scope of this initial proposal. We do know that there are other biomarkers available that are probably more effective in certain circumstances, and it's something we want to address through the second phase of research.
Okay.
So can you hear me? Okay.
Yes.
So I'm not sure if you're aware. I remember a few years ago there was a iGEM team project. I don't remember the name, exact name, but I think the nickname is like a colorful poop, right? So they have similar ideas before, and instead of using JP as a reporter, why don't you just go one step further? Once you see something, let the cell lyse, then release some agent that show different colors, right? So you skip one step using the black light. You can just turn around and look at it. Then you know, right, what's going on. So that's idea sounds very similar to this one, right? And also there are Chris and Ben brought up the issue, and you don't even know whether the CA 19-9 can be sensed, right? So I think if you want to go this route, you need to do more research to at least first to show it's feasible, and second to distinguish this project from that one.
Also,
Yeah, that is a very,
to build off.
oh, sorry, go ahead.
Go ahead. To
just
build off that point. Okay. So Mikhail Shapiro at Caltech designs ultrasound reporter genes that he expresses in E. coli and other gut bacteria with the idea that you can do this in a living person or an animal. So you would just give you give an enema of the E. coli and it would kind of seed the intestines, and you do like an ultrasound test. And not only would you be able to see increased levels of response, but actually like focal point. But again, that all hinges on having a sensor for this marker. So if you can find that, if you can find a receptor that interacts with C19-9, then you're off to the races. But that's the key point that has to be solved here.
Yeah, I do believe that this is a very important distinction to be made. The other project that you're referring to was iChromI, and talking to some of the people who will be judging for the iGEM competition, they believe that the similarities are actually a strength because we can learn and improve from their failures or their places take up where they left off. Again, when it comes to the sensing, we definitely would need to do more research. That is definitely the largest pitfall that we have to overcome for this one.
And to build off what Savage said, one of the examples of us building off the eChromI procedure is their method of delivering their E. coli or whichever bacteria they used was with a probiotic drink. And we were examining that route, but we were worried that the traveling through like your stomach and the esophagus and all of that would damage whatever alterations we made to the bacteria. So that's why we went with, altering to like using the suppository, and basically intend on making improvements based off comparisons such as that.
That problem's been solved. They do make coatings that you could take a pill and they don't open until they get farther into your intestine. So that one you can get around.
Another hope is that this might be more long-term than the single-use drink that they came up with. Theirs was a very much a in-the-moment diagnostic test, while we hope that the suppository method might end up being a little bit more long-term effective. While we don't believe it would last a lifetime for a person, we believe it might be able to last longer than the immediate test.
In the chat, I put 2 possible receptors for CA19. I don't know too much about them, but these are possible receptors that do exist, and we could look more about them. If they don't exist, we did, I think we gave other antigen like CEA, which is more common. I guess we can look at that, but I think more research needs to be done. But I think that research does exist, and we could possibly do this.
The Siglec-7/9 receptor relies on this marker to actually be attached to the cell, so if it's attached to a surface protein of a cell and it interacts with it, then that receptor has a mechanism. But it doesn't mean you can't, it does have a binding domain, so it would take some engineering to do it. Unfortunately, getting surface binding working on E. coli is a real tricky problem. It has an outer membrane and things tend to not translate from the outer membrane to the inner membrane. So I could see this as a eukaryotic cell assay, but you would have, using this Siglec 7 and 9, but you'd have a hard time adapting that over to E. coli. But the binding domain is there, so that is something you have to work with. I don't know what the specificity of those domains are. You have to look that up.
Yeah, no, I agree. I think the fact that there is something in mammalian cells is a good first start, but then it'd be nice to find something in some sort of bacterial species that would function similarly.
Yeah.
But access to
Dr. Wang, do you know of any? Does anyone know signal transduction from outer membrane of E. coli to gene expression? I don't think I've seen that.
So
Maybe.
that I'm not sure, because they talk about chrome sensing, right? Chrome sensing molecules typically just go through the membrane, right? They bind to the protein, they induce the transcription. And in E. coli, I never worked with those kind of a transmembrane kind of like a receptor thing. That's more common in mammalian cells, right? Chrome sensing molecules, typically they're small molecules. Just go in and out, right? They don't need a receptor. So that's another thing. But they just, who knows, maybe you can just, one thing I've recently do some research is they see bacteria is co-evolved with human for billions of years. They might evolve some protein that can sense something, but you need to dig it out.
Also, I guess this is a sugar. So if it's small enough to get into the paraplasmic space, then you have access to the inner membrane and things get a lot more simple. But we'd have to look that up. Usually only small metabolites get into the paraplasmic space.
Yeah, and I think the delivery method also is going to be an interesting thing to sort of work on as well. Because whether you'll be at actual, like the bacteria to maintain an infection of the gut is a question, because everything is moving through that system and going out, generally speaking. So whether you'd be able to get that to actually stick and stay is a question. But I think that is something down the road. I think having a receptor, I think, is going to be the key next step. Don't you guys agree? Like, and then how that you link up to some sort of transducing that signal into the gene expression is like the next thing, right? So, okay, cool.
I hope that was it, because that's 10 minutes. And now if we can move on to the grass team, I think you guys wanted to go next.
Yeah.
Okay.
Yeah. Before we start, I'd like to mention that Marissa was an integral part of our group, And unfortunately, the only reason she's not here right now is because she has work.
Yeah.
Okay. Now I will begin.
All right.
Okay. Okay, take us away, David.
I don't think we can hear you.
Yeah, David, you good?
I hope you're telling us why that's not a picture of grass.
Yeah, it's we're working with it. Okay.
I was a botanist for 2 years. These things kill me.
Yeah, no, we know for sure. Okay.
Okay, I think maybe he just got kicked out of the meeting or something. Bad
Yeah,
internet.
he may have gotten kicked out. Would you like to wait for him or should I begin?
Just start, or we can have water go if that works. You can just, I'll let you go.
Okay. Okay. Sorry for the technical difficulties. Yes, we know this is not grass, but bear with us because the content is more important than the image. So as you know, we are the Grass Project, and today we'll be telling you about our idea. So basically, we noticed that Arizona uses an inordinate amount of water. It's primarily a desert landscape, and yet people insist on having lawns, golf courses, public parks covered in grass. And it needs constant watering and reseeding, and up to 70% of residential water use is used outdoors for maintenance. So how can we reduce water consumption while allowing Arizonans to maintain this beautiful aesthetic that they love and keep their golf courses and become more sustainable? Our solution is to engineer a plant microbe and affect the root rhizosphere of Bermuda grass to increase its resilience to drought and in hot conditions.
So the microbiome is incredibly diverse, and it can constitute basically most of the plant. And in reality, what we see here is a complex interactions of many different microbes, most of which can be just labeled as the rhizobacteria, or some of the bacteria that are found in the root system of our plants. And a lot of the research into this section has found that most of these microbes can help contribute to the drought-resistant properties of certain plants, as well as supporting growth and resistance to other stressful factors, including abiotic and biotic stresses. This could include the uptake of nutrients, improving that, as well as resistance to drought or high temperatures, low temperatures, as well as other pathogens or potentially even fungi. And the ability of certain microbes to produce biofilms within your plants can improve these capabilities even more. Next slide. So we have seen in some previous teams in iGEM that there has been focus on both editing plant genomes as well as the rhizobacteria. And while it is possible to go through both of these routes, we've decided that focusing primarily on the rhizobacteria would give us an easier access point to editing and producing noticeable changes within our plants. And this is primarily due to the fact that editing the plant genome can be a little complex. They have multiple chromosomal copies. Some of their sequences are incompletely mapped. And then there is the possibility of some self-infertility within plants that make it a little difficult for breeding to occur within your plant subjects. So focusing on altering the microbes within the rhizosphere is definitely one of the high points of our research. And of this, modifying and looking at biofilms presents one of the more interesting options to sort of enhance our diversity, as well as increase the production of certain enzymes and the flexibility and solubility of certain polysaccharides, including alginates and exopolysaccharides. And by focusing on altering these target chemicals, we can hope to essentially increase the absorbance of nutrients as well as water and maintain these important factors while our plants are under stressful conditions, so as to avoid or inhibit senescence.
So this is going to be, of course, beneficial when we are successful. If we are successful, this will be beneficial in reducing water consumption, which will reduce cost for individuals. But we're not blind to potential risks that we may have to face. Is increasing the grass's hardiness going to make it like an invasive species and take over, someone's lawn? Will our new bacteria be compatible with the existing bacteria, and would it be toxic to other animals or fungi in the soil that's so necessary? These are very important questions that, of course, we will address. But we also see future applications extending into farming and agriculture, reducing water consumption for other plants, and allowing people to have lower maintenance grass that will be healthier and longer lasting, also reducing the effect of excess water consumption in that area. So this all sounds great. So how are we going to do it? Well, basically, we'd like to get native bacteria. So we would get our bacteria from the creeping bentgrass rhizosphere, which is native to Arizona and does recruit it. And then after we've isolated it, we would design a vector that would promote EPS production. And we would tag it with GFP so that we can verify that we've been successful in integrating this plasmid into our bacteria. So next, we're ensuring that it's been taken up. We would use qPCR microscopy to ensure that it's actually in our bacteria and will be successful once we implant it in our soil. So we'll inoculate our seeds, our Bermuda grass seeds, some with unedited bacteria, of course, and some with our new and improved bacteria, and leave them ideally in a greenhouse, which ASU does, have some of, and monitor their growth. So how will we know if we've been successful? Well, we'll need to take a cutting of the rhizosphere of both groups and determine through fluorescence microscopy if the bacteria we intended has actually formed the biofilm around the roots and is actually the one causing these changes. We would also do a weight comparison of the roots from both groups of grass. This is a method that has been used before to determine if the water uptake has increased or reduced based on the weight of the root. We'd also quantitatively measure both grass groups in terms of plant growth, height of our grass, and qualitatively measure the color and, the wiltedness and overall aesthetic of both grass groups. Now you'll ask, what if we fail? This is a very real possibility that happens all the time. We have a few potential pathways that we could take if that's the case. Is designing the vector too complex? Are we having difficulty isolating our data? Perhaps we need a different vector that doesn't speak to the EPS production. Maybe we need to focus on alginates. Maybe we need to double check our control variables and ensure that we can properly measure what we're looking for. And then we'll consult with our excellent professors and our lovely graduate advisors and try again. We have multiple pathways.
And looking through available vendors, we were able to calculate a rough estimate budget based off prices of each product and also maybe how many times we will have to use them. So for example, as the main thing with the organisms, so the grass seeds, the isolated strains of the bacteria in the soil, that could range from about $200 to $300, and this is rough. And it could be more or less during the actual experimentation. And then when it comes to the technology, much of the lab/tech equipment would be available through the ASU labs. So really, this estimate of about $1,700 to $9,600, I would strongly believe that that most of the machines would be available. And if there's really any that's actually needs to be purchased, it would be probably like a few things. When it comes to funding and all that, that could be provided through our outreaches. So to fund our project, there are some grants we could apply to. We have the USDA's Conservation Initiative Grant and EPA's People, Prosperity, and the Plant Grant for Technological Innovation Sustainability. In addition to grants, we will need the support of others to make impact on our community, such as sponsors and help with Changemaker Central, ASU's Resource and Innovation Network with the School Sustainability, and the organization Project WET that seeks water security for the future. And also, if we can bring together our DIYbio team, sponsors, and money to create a successful project, we would envision our grasses being implemented throughout the metropolitan area. But our first steps would definitely be Emma's backyard and hopefully perhaps her neighborhood. Plus, we are going with a school with #1 innovation status. So, of course, ASU can take those water guzzling lawns and implement our winning grass.
That's all from us. Thank you very much.
So I just want to clarify. So what your team is trying to do is to use subtilities to promote the expression of EPS, right? And which in turn will help the grass grow, right?
Yes.
And so there are transporters for EPS in the grass.
Absolutely. EPS is actually already a product of this bacteria, and we'd like to upregulate and increase the amount that it produces, or potentially knock it out and see whether that's going to significantly affect our grass's success.
Has someone ever do you guys have a study where someone might have supplemented with like EPS just directly into the soil to see if that increases plant growth?
I don't have a catalog of all of our research in my head right now. I would have to look back through and verify. I'm certain there's something out there that we would definitely find if we haven't already.
Okay.
I think most of our studies were comparing just subtilis that were known to upregulate EPS versus subtilis that weren't. But that's all I have in my head right now.
Yes, so actually there are several options that are available when it comes to expression of EPS. We've mentioned that inoculating our seeds directly would help promote the bacterial growth within the root system. And that has been actually tested, and results have been seen very positively, especially with diverse microbiomes. However, we have also seen some literature where they do focus on just the soil environment and introducing new microbiomes into a soil, as well as actually sort of like a surface covering. So they've actually sprayed their leaves with different inoculum in order to produce like EPS or other sorts of biofilms.
One issue that I have seen with microbiome engineering for plants is that some forms of B. subtilis are not compatible with some forms of grass. And so I'm concerned that you're taking a B. subtilis from a non-Bermuda grass and then trying to translate that over when you could just be taking a known B. subtilis strain that does interact with Bermuda grass and engineering that. But I also wanted to point out this is a really great approach because the regulatory space for engineering the microbiome of plants is not regulated the way that the plants themselves are. And so you should actually not have a major problem with implementing this. Whereas if you were going for the plant itself, you'd face a lot of regulatory work with USDA. So that part I think really has legs on it. I would warn you to look at which strain you're going to start with to make sure you have that optimal compatibility.
And what is the life cycle time for Bermuda grass? That was one thing I was wondering. Do you guys know?
I know that the B. subtilis bacterium will localize to the roots first, so we would be able to gather some information after, like, as soon as the roots have formed before it's fully formed. I think our original plan said 30 days in a greenhouse growing in order for them to sprout. I would definitely double check that before we begin.
Yeah, because I think, you know, depending on what kind of experiments you're planning, that can be a big thing. I mean, I think you are kind of aided a bit by the fact that you're not trying to genetically modify the plant itself, right? So again, same thing. You can probably do lots of different experiments almost simultaneously just by modifying the bacteria, and then yeah. But it's something to think about, because if you do have to grow up lots of seeds, then that can take time, right? So
Fair enough.
Yeah. How well described is this process of forming a biofilm? Is it just known that those are the chemicals involved and those are the enzymes, or has this been pretty well mapped out in this bacterial strain?
So I do know when it comes to a lot of the biofilms in the studies we were looking at, it's not just one bacterial strain that's involved. There's usually a cocktail, and so that means there's a variety of different factors in play. So I don't think we have a significantly mapped out process of that currently within our research. That doesn't mean it's not out there, but that's not something we really have specifics on yet.
What we do have a little bit of knowledge on is the actual transport mechanism and the production of EPS. And it is a little complex. There's plenty of different genes and enzymes that are involved in the whole process, which is why we have initially considered focusing on targeting key enzymes in that pathway in order to see a difference in production. But we instead wanted to sort of test out how can we sort of look at the microbe as a whole, and how can we implement that whole biofilm system into our strain or grass.
I'd also like to
So
mention that.
it already,
Oh, sorry. This is in response to your other previous. I also mentioned, taking a root cutting and using fluorescence to determine if the bacteria that we intended is in fact localizing to the roots as we described. So to me, that's also another step that we could take to ensure that this biofilm that you just asked about is actually the one on the roots doing the work. So we would absolutely have to verify that. And I think we can.
It seems like there could also be a simpler approach that doesn't require engineering, which is just to take the microbiomes of drought tolerant grasses and see if you can seed Bermuda grass with those same things, right? But that could be an early stage experiment that doesn't actually require genetically engineering B. subtilis. Just throwing it out there.
yeah, I mean, because the thing is, right, that at the end of the day, the biofilm is going to be made up of not just one organism, it'll be made up of multiples. And the question is, what's going to be there in the soil already, right?
Mm-hmm.
So if you stick, is B. subtilis already there, right? And what B. subtilis is there is kind of one of the questions, right? Because that was one thing I was wondering is, making the biofilm is one thing, but then making it in the kind of soil that's around here is the other question, right? Right, like what's different about the soil we have here than say somewhere else?
Okay, yeah, well, this also kind of feeds back into, I think it was Dr. Bartel's question earlier of, why are you taking, one kind of bacterium from one root and moving it to another root? That I would be hesitant to include the biosphere from, a completely water independent plant and just trying to transplant it, although it could be a preliminary attempt. I think this could allow for individualization, specificity of which plants you'd like to, become more water resistant.
Okay, unless anyone has like a dying question, that's all the time we have right now.
Thank you for your questions.
So, Good job. Both cancer and grass. Someone from water, excited to share their screen.
Okay, I'll give our little introduction. As you guys know, we are investigating, sorry, let me turn off my notifications. We are investigating contaminated groundwater on tribal land, specifically up north the Navajo Reservation that mostly is in Arizona.
Okay, so for some background, there are approximately 521 abandoned uranium mines that have made the groundwater on the Navajo Reservation in Arizona undrinkable. And the contamination from uranium is linked to kidney disease, lung and bone cancer, pediatric neuropathic syndrome, and a whole host of medical problems. And also a lack of clean water poses an issue for, oh, could you go back to these? For basic sanitation, and also, especially during the pandemic, it complicates a lot of people's living situations. And so for most families on the reservation, the closest regulated water supply, i.e. clean, is about an hour's drive. And this is like the minimum. And so because it's so long and it's quite an obstacle to drive an hour, a lot of Navajo Nation residents get their water from unregulated sources or water supplies that put them at risk for getting radiation-related disease. So that's a pretty big problem. And on the next slide, I have just this map to show you how many uranium mines there are, and these are abandoned and they pose significant risk to the water sources nearby. Next slide. And so for context on the science behind groundwater contamination, it usually happens with uranium because of waste piles and open pit mining below like zones of saturated groundwater produces mine wastewater. And this increased exposure to water and oxygen mobilizes uranium and arsenic, which causes contamination of the water. And arsenic is a naturally occurring contaminant in the southwestern groundwater. And so when people mine for uranium, it increases its concentration because high arsenic and uranium concentrations occur together because they're more soluble when oxidized and are likely to be co-precipitated as minerals together. And so that's why uranium and arsenic are usually found at really high concentrations in areas associated with previous uranium mining, which is why they pose such a risk to families on the Navajo Reservation.
So based on that, our goal is to engineer a system that transforms both arsenic and uranium in groundwater to less toxic and more easily filterable species. However, our solution won't actually involve the filtration process, but it will make it more possible to actually filter out the arsenic and uranium. So there's three main ways that we've come up with that we can do this. The first is to clone the arsenic oxidizing genes into a bacterial species that has uranium reducing capabilities, whether those are natural capabilities or they've been engineered into the bacterium already. And then the second is to kind of just do the reverse, where we're cloning the uranium reducing genes from Shewanella onadensis into arsinotropic proteobacteria. Or we could clone both of the uranium and arsenic genes into a model bacterium like E. coli.
All right, so I'll start with kind of the science about previous work in arsenic removal. I read this review of arsinotropic microbes. As you can assume, an arsinotrope is a microbe that is able to fix arsenic. So arsenite, which is the arsenic 5 species, is less toxic and more easily filterable than arsenate, which is arsenic 3. Many bacterial species have already demonstrated an inherent ability to make this transformation. For example, in E. coli, they have an arsenic operon that reduces arsenic 5 to the more toxic arsenic 3 just to get it out of the cell, and it has its own transporters for that. But we kind of want to do the opposite and instead oxidize arsenic 3 into arsenic 5. And to do this, there have been genes identified in proteobacteria. The AIO gene specifically is an arsenic 3 oxidase that's able to transform it into the more easily filtered arsenite. And similarly, there are homologs for other bacterial species. And just one example, the Ralstonia genus has been shown to have an inducible AIO system. In that same vein, arsenic oxidizing bacteria in one study were just like various strains were collected from it was called active sludge or something. And they tested all these strains for oxidation efficiency. And they found that pH of 6 to 8 and temperature of over 20 degrees centigrade is optimal for this oxidizing activity. And just to touch on a previous iGEM project regarding arsenic filtration, since bioremediation is kind of a hot topic at iGEM, this group engineered the constitutive production of metallothionein, which is a metal binding protein from this E. coli ARS operon that basically allows them to extract the arsenic out of the cells. They also coupled this to the removal of cobalt. So it's not too different from ours, although uranium is a little bit trickier than cobalt. So then to move on to uranium. So as I mentioned, many proteobacteria are capable of doing arsenic oxidation. There is one beta proteobacteria called Burkholdia fungorum that's capable of doing uranium respiration. So like we kind of mentioned earlier, this doesn't entail the physical removal of it, although it does make the uranium like possible to be removed. The soluble uranium-6 is the oxidized form of uranium. But once it's reduced to uranium-4, it's insoluble and able to be, removed out of the wastewater. So that is a possible model if we chose to, go the route of like adding an arsenic phenotype into the bacteria that already does uranium respiration. Furthermore, there are a couple bacterial species that, while they don't reduce the uranium, they are able to remove it from surrounding water just by taking it in, and these cells can later be washed with EDTA and the uranium harvested. So that's another route, just to directly, absorb uranium into the cell rather than have the cell do that kind of reduction and spit it back out into the water for subsequent retrieval. Finally, Griffin, one of our grad mentors, shared with us an article about a startup using genes from this strain Schuanella ondiensis. And basically, in this kind of startup idea, they are taking a resident fungi from Chernobyl and using these uranium-reducing genes from this bacterial strain to just kind of do some radiation cleanup over there. So there's definitely a lot of work to be done still, but there has been demonstrative work regarding uranium removal and reduction. Similarly, there was a previous iGEM project. Although they weren't extracting uranium, they did come up with an adsorption technique to detect it and harvest it. So we were thinking that could kind of be a launching pad for, the second step of physically isolating these toxins once they've been, transformed to their insoluble species.
Sorry, I was muted. All of our routes we propose require the same materials, but we didn't put a price point on them yet until we knew exactly what sort of bacterial strain and plasmids we would need specifically. But just general materials required are going to be plasmids, primers, the bacterial strain, enzymes, chemicals. We'll need arsenic and uranium. And then in order to work with uranium on campus, everyone involved will need to complete a radiation training course. Not sure how extensive the course is, but it is a requirement in order to work with it. And then the general timeline for the project would be just cloning the genes of interest into the plasmid and then confirming the gene was inserted, and then transform the plasmid into the cells and then confirm that plasmid was inserted. And then we'll need to test the transformation efficiency of arsenic and uranium. And then a previous iGEM team used flame absorption spectroscopy. Sorry, and I'll just finish this up real fast. And then we'll need to develop and apply an absorption technique to extract, reduce arsenic and uranium. Let's see, and then can we just go to the next slide real fast? And then these are faculty who could potentially help us with our project on campus.
Our last slide is just the elevator pitch, so it's just a summary.
Awesome. Great job. 10 minutes. I know this is going to go a little bit over, but I want to be respectful of your time. So 10 minutes for faculty response and questions.
Can I have a question?
Yes.
So, it's good job you mentioned the previous projects and both for arsenic and the uranium. And just can you again just highlight your novelty and distinction between your proposal and the previous projects?
Right. So as far as the previous arsenic group, their goal was to filter out arsenic and cobalt both. So it was kind of similar in that sense. One of the advantages of kind of building off of their project is they already have a plasmid. However, the route they took was to produce methylthionine, which wasn't something that was totally on our radar. We were hoping to use like an oxidase or something to oxidize arsenic rather than bind it to a methylthionine protein that would then kind of, aggregate and precipitate out. So I think that's kind of the main distinction between our project and the previous arsenic project.
How about the uranium one?
And the uranium project was just a detection project. It was more diagnostic. They engineered a bacteria that basically made an adsorptive biopolymer able to extract the uranium.
So your team proposed to do both, right?
Yeah, our proposal is to just fix both arsenic and uranium in contaminated groundwater. And then subsequently, we would have to build off of one of their techniques to not even their techniques, but any technique, just to then filter out those insoluble species.
So I worked with a lab a while ago that basically was doing a little bit of work on was something that you reduced uranium, and I totally forget what it was. I think it came from like the pipes of like oil pipelines, but basically I think a lot of those require them to be kind of in and I don't know if that's the case for some of these, but basically they require to be in anoxic environments, so no oxygen, right?
Right.
And so I mean that it has its own fun things that you have to do. Are these bacteria going to have to live in an anoxic environment as well?
Right. So the one bacterial strain I brought up just for its uranium reducing abilities, the Shewanella ondiensis, is able to reduce uranium both aerobically and anaerobically. So if I don't know how, transferring genes from the bacterial strain to a different model would affect the function, but we have that going for us.
Okay. Yeah. Because I think, just in terms of feasibility, that makes it a lot easier if you don't have to basically get rid of oxygen from everything before you do it. Right? Yeah. So, okay. Yeah.
One issue that these approaches continually face is that they reduce the cost, but they don't reduce the necessity of having a water treatment system. Like you do need to have sediment ponds, you do need to have some sort of filtration, and it's not something you can, I think the dream is you have an arsenic laden well and you inject it with some bacteria and then it cleans itself up, right? Precipitating out all the arsenic and making the well water clean. And I think that's where a lot of these have these projects, because there are others, they fall apart at that stage because you still are asking people to build infrastructure, and then the current infrastructure is already there, right? There are ways of doing this industrially, and so method. The one way around it, it would be the formation of nanoparticles that incorporate the uranium. So one example of this are the manganese nodules that are, so the bottom of the ocean in the Pacific is covered with these manganese nodules that are actually a bacterial community that oxidize manganese into an insoluble form and form like a matrix that they crawl around. And so people have seen this for other metals as well, including tungsten. So something like that would get around the need for having a waste treatment plant on site. So just bringing that up, as you're choosing genes, the ability to form an insoluble, not just precipitate, but an actual particle. And bacteria are quite capable of forming nanoparticles. They form iron particles, manganese particles. These are all normal things bacteria like to do.
With uranium, it might get dangerous if you put too many of those together, right?
Yeah, that was kind of the other thing I was wondering about, because I couldn't really find much, doubling up these kind of pursuits.
Oh, most of it's not that radioactive.
I know, I'm just joking. You'd have to get a large amount. But I think like if there was a way even to make some sort of a biofilm or something that traps it, could potentially be another way, right? Although that might break down pretty quickly unless it was somehow structured. So yeah, but I think that's actually a good consideration.
Yeah, I was just going to say, to be fair, it kind of was something that, it was a problem that we faced like halfway through our research, like, oh, this really isn't filtration. Like that's not really like accurate. That's kind of the subsequent step. So that's not something that we've really put too much focus into yet, unfortunately.
But it could be also used at the site, right?
Right.
Because you could see infecting or like basically scattering these bacteria over top of like tailings. And then if that makes it insoluble at the source, then it's not going to travel potentially as far, and which then that does sort of have the effect of making. Yeah, I mean, you could see that also being one possible sort of way to use a system like that.
As more of a preventative.
Yeah. Yep. Stop it at the source, but I mean, it doesn't help with the stuff that's already out there, right? So.
Right, the idea could be to seed the current, I guess they're called tailing ponds, with that to stop it from spreading, which is a huge problem.
It is. Because there's still tons of it there.
Yeah.
Just waiting to be released. So.
It might be worthwhile to harvest from those ponds. So the way people find resistance to pesticides now is they just go to the pesticide factory and look in the soil because it's so laced with the pesticides. Anything that lives there has to be resistant. So it might be worth a fishing expedition to pull out what organisms survive in these high uranium environments. They usually have a sequestration method. And so one gene in particular would be the paraplasmic binding protein, which it's usually what grabs onto these small ions and then we'll either bring them into the cell or like sequester them somewhere else. And I know that there are tungsten and gold specific PBPs. So you could do like a genomic fishing expedition and say, all right, well, here is it enriched for some PBP-like protein. And then by cloning that out, you may be able to find a uranium-specific sort of scouring molecule, if it hasn't already been found. I don't know, I haven't looked for it.
Right. Well, I was just going to point out one of the papers I read kind of took a similar approach. They when collected samples from activated sludge was, I guess, the technical term for this, but identified Bacillus and Lactobacillus as two like bacterial species that are pretty apt at removing uranium just through absorption.
That's lucky because those you can work with those. The
Right,
problem with
that's
a lot of these is we don't know how to culture most bacteria.
Mm-hmm.
Okay. Good job. Thank you, Water. At this point, I was going to separate the professors into their own breakout rooms so they can decide on their rankings and come back and give us the faculty choice awards. So I guess I don't have like a time limit for you guys in there, so I won't close it on you, but just come back when you've come to an agreement, I guess. Does that sound like a fair deal?
Deal.
Okay. I mean, ideally don't take longer than like 10 minutes would be nice.
Yes,
but okay. That should have opened. Okay. Well, now that they're gone, so, if you guys have any ideas for like the other projects, like David, I know you had one. That'd be a cool discussion to have. So, opening the floor.
Well, first of all, good job, everyone. They were all great. They were all great presentations.
Yeah, great job. It was very evident that you guys put like a ton of work into this. So, very happy to see all of them. Very cool.
Yeah, it was a very intense questioning. I think it was really good for all of our projects, but oof, that was something else.
Oof, indeed.
Sorry, I missed the first two, or I caught like the last end of grass project, but yeah, how is this? Are they like choosing the project, or are they just giving you the recommendation?
They're doing like a recommendation type thing. The first place winner that they choose will get a little prize. It's not
Okay.
It's pretty fun, but it's not anything big. And then the students are actually gonna be the ones
Yeah.
choosing. Which I have.
Yeah. What I saw, everything looked pretty good. But yeah, I mean, of course, there's always improvements, but that's pretty easy to go through.
David, did you want to ask your question that you had at the beginning?
I think it was about, if I remember correctly, yeah, it was the CA919 sugar. Was the thought for you guys in the cancer detection group that you would have CA919 bind to a receptor, and then from there, that would increase production of whatever molecules were needed for quorum sensing to then start the process of producing GFP, or am I completely missing that one?
No, you're pretty close to the point. After getting some really good feedback from our advisors, we decided that modifying the quorum sensing system would be the most successful and the least technical of all of the options available. We planned on finding a receptor for the CA19-9 antibody or antigen, and then placing it replacing it with whatever anticodon or anti-encoder, I think is the technical term for the quorum sensing. Replacing that with that receptor that we found, and then just have that connect to GFP protein.
Gotcha. Thanks for clearing that one up. Yeah.
Yeah. For the water treatment, and I guess the grass one too, I had a couple questions for you guys in regards to what you guys were considering in terms of ensuring that whatever microbe that you find, test, and then release wouldn't be disruptive to the environment that it gets released into.
I think, at least for our group, that kind of goes along with what I think it was Dr. Bartel pointed out at the end of our presentation, to select a bacterial strain that's already like native and shown to be present in these areas, I think is a good starting point, and working within those rather than bringing a non-native species in.
Okay, cool. Because that's definitely the process we were planning on doing with the cancer one, for the microflora. We decided,
Right.
oh, let's just modify something that's already there.
Yeah, it makes the most sense to me too.
I think something to, sorry, you can go.
I was just going to say for the grass team, we can, that's something we considered in our human practices. It's also why we're extracting a local bacteria. And honestly, like we would just have to expose it in a lab setting and make sure that it, that we're okay.
I think one of the biggest things I see in projects is considering a kill switch. So that way your recombinant bacteria isn't spreading throughout the environment. So like if there's a certain metabolite that's only present in whatever environment you're using, you can make that cell dependent on it. So that way, if that metabolite disappears, it dies. Or like vice versa, where if it does get exposed to a metabolite that is present around, like normally around, but not in that environment, then it gets killed. Or, I mean, there's a lot of other types of kill switches and sensors you could use. But I'm usually partial towards projects that talk about kill switches because it's just a basic thing that I think teams should always talk about. But yeah.
I had a question about the voting. Just about, I know that we, like the students also, we do like something about ranking.
Yeah.
I just was curious about how that was gonna work.
So I'll actually send it now. Don't fill it out yet, obviously, but let me know if you guys can access that. It's a Google Forms, and it's just gonna be how we're choosing it. You guys are gonna rank your first, second, and third choice projects. You can choose your own, but like I said earlier, I mean, winning or losing this thing doesn't really matter for like if you get on the team or not. So really do try and vote for like what you think is the best project. Put on your iGEM hat, not your cancer, grass, water hat, even though they're all very cool. And then the faculty vote kind of thing. #1 innovation. There's going to be like a little prize, but it's not basically just it's so they can provide more feedback. Your cancer, grass, water, yes. So they can provide more feedback for you, and like so you can take as much of that into account as you want. Ultimately, you want this to be a project that you're excited about and can spend a summer working on. So yeah. But did that Google form, like, could you guys all access that? Yes. Okay, cool. So yeah, I encourage you to ask a lot of questions because you're the ones deciding what we spend our summer on.
I had a question, Margaret, about the logistics. How much, so funding is going to be a major aspect of this. I know a big part of our funding is going to go towards transportation fees for the actual competition itself. But for the research, is that something that we as a team, like everyone, will have to reach out to funders about?
So about the funding for the travel, we're still working out like the budget and how that's going to work because the competition's in Paris. We're going to see how much of travel we can actually fund. But in terms of actually reaching out to sponsors and whoever, that could be something that we make a team effort. But yeah, honestly, not sure yet. Great question. But that's definitely the next step. We can't really ask people for money until we have an idea. So that's what this is for. But yeah, we could totally make it a team thing to reach out to vendors and stuff and at least gather the materials to ask. That's kind of why I asked for a team picture at the beginning of this, So we could have something where it's like, hey, this is everyone who's been working on it so far. Even if it's a Zoom group picture, I think it's nice to have. But hopefully that answers your question more or less. Everything's kind of up in the air right now still, especially with COVID.
Yeah, I totally get. With regards to the water team, I know you guys wanted to avoid finding specific numbers in terms of like how much it's going to cost to finance that research. But do you guys have an estimate?
Yeah, to be honest, we kind of like last minute, we're doing the materials and stuff, and so we were like, okay, well, we don't have a specific route to go with yet, so we'll just not bother looking into it. I did look that one strain that I kept bringing up, the Schuanella onwaniensis, or however you say it. That was about
some biology name.
Yeah, some biology name. That was about $400 for that strain. And then plasmids, I again, it kind of is dependent on what plasmid we're looking for, but from what I could tell, I was looking at one the other day, and I think it was like 130, 150, something like that. Primers obviously aren't too expensive. Enzymes, we probably will already have mostly, just, restriction, ligation enzymes.
You guys know you get 30,000 free 30,000 amount of
I didn't tell them that. No.
Yeah. So when it comes to plasmids and DNA synthesis, you guys get 30,000 base pairs of free synthesis from IDT and I think it's Twist. IDT does 20,000 and Twist Bioscience will give you 10,000 for free. So you guys don't need to worry about the cost of gene synthesis.
Right.
Okay, so I think we had a discussion, and maybe Chris can give you the ranking later on because I have to go. So I will go first about my two comments. The first comment is I think this is applicable to all three projects you guys proposed. It's all great, very creative, excellent ideas, but I think I'm maybe too negative, right? The two suggestions I want to bring up. Number one is all of you propose something. The designs to me at least sounds too straightforward. These things helpful, beneficial. Let's produce it, right? There's no more like a so-called computation or processing behind it, right? If you're sensing signal, if the signal is high, we do this. If the signal is low, we do that, right? Some kind of a computation behind it. Or if you're sensing signal A, we do this. If you're sensing signal A and B, right, we do that, right? You read the papers, you saw this AND gate, there's circuit, there's fairly complicated design can be incorporated into the circuit. And that's not, I'm not saying that in any of your design, right? And the second point I want to raise is about being quantitative, right? It's engineering. You have to be quantitative, right? And when during the discussion, I mentioned to Chris and Ben, right? You cannot expect you produce something, the magic happens, right? There are quantitative aspect to it. You need to plot a curve, experimentally measure a curve, right? If the signal is low, or if the gene expression is low, this happen. If gene expression is high, this happen. If somewhere in between, something else is going to happen, right? To be a little bit more quantitative, both experimentally, maybe all of your team need to recruit someone who can do some kind of modeling or dynamic measurement or modeling, right? So those are my two comments. Okay, sorry I have to go, I have to leave early, but oh great. I think Chris and Ben will be here and announce your ranking. Okay, bye-bye, have a right.
To echo that point, a lot of these things come down to the concentration of the thing I want is this, and the binding affinity of the thing I want to detect is this. And if they match up, you can go. If they don't match up, you've got to keep on looking. And that holds true, that's super, super important for biological detection of a ligand. So if the plasma levels of your biomarker are below the affinity of a receptor, then that cannot work. There's all kinds of metal binding proteins out there. Uranium binds to everything. It's actually used in EM because it provides a nice even coat, but it does so at a very high concentration, and you need to find it at a low concentration. So being able to say the average concentration in a well is this, but we know that the affinity of this enzyme is below that, so we have a reason to believe it's going to work. That's what convinces people to move forward, because everything boils down to mass action. If you talk to the old guys, old guys don't believe in synthetic biology. They only believe in mass action. It's all just chemistry. Everyone smiling has heard that one already. So.
Okay. Yeah, and I basically have little to add to that. I mean, I think and I know that basically the goals of this were to try and sort of start to figure out the viability of these ideas, and then to start to think through the process. And so I think you guys have definitely started that, but I think I just echo those things, is that, think about how you're going to do this, right? Like very deeply, because that's where, it's going to make or break any of the different processes that you guys are planning. So, but I think you guys did a good job presenting, and I think you should give yourself a pat on the back, because I think all of you had very coherent presentations. I think the information that wasn't there was just what they were talking about. I think, we don't expect you guys to know all of that off the top of your heads, and you're all learning. I think that's a really important aspect to take away from this as well. So, so let me give you our rankings, okay? So number 3, we have cancer, okay? The reason for that was just that a couple of the different aspects of the model weren't there for us, and we think that is going to make the viability somewhat more difficult to determine, right? But I think the idea is obviously a reasonable idea, but I think, like we were talking about, where's all the information about, like, what receptor are you going to use, right? What is its affinity for that kind of a ligand, and how much of that ligand do you expect to find in someone's stool, right? All those kinds of things are super important for figuring out whether that's going to work. And then we actually are going to give a tie to the other 2 groups. We thought both of the projects were very interesting and had some cool aspects. And I think, from our point of view, we think, with further development, like either of those could be potentially reasonable projects to go after. So that was what we did. So good job, everybody. And I think, as you're moving forward, if you have questions for us about the things that we were talking about, I'm sure we'd be happy to come back and give a little bit more information. And also, like, I think, finding and getting a mentor here that if we can get you guys hooked up with the right mentor, I think is going to be a big help for that as well, right? Because they're going to be the experts in those systems, and hopefully they can help to sort of work out some of those details. But I think, yeah, good job. So.
Yay. Good job, guys. There was supposed to be only one team who won, but that's fine. The 2 winning teams get a fun little, assuming you come back to campus, a very cute little 3D printed cookie cutter with ASU. So now we'll have to make more of those than originally planned, but that's fine. Congratulations.
PLA is not that expensive, so we'll be good.
We did that just to mess with you, Maggie.
Just to
We'll
mess
still have
with
to
me.
make extra. Sorry.
I actually think I volunteered to print them, but we'll see. So.
Well, now I will hold you to that.
Yeah. All right.
Yeah.
Cool.
Otherwise, I don't want to keep you guys too much longer. So.
Yeah. Well, great job, everybody, and I'll see you all soon.
Professors are free to go. Students, I mean, you still have to be here for a little bit. So, thank you guys, Dr. Bartel and Dr. Glazer.
Bye. Thank you, everyone.
Thank you.
For the rest of you, You're welcome to, I noticed someone already filled out the form, which is fine. But for the rest of you, you're welcome to ask questions of each other or just fill it out. I'll leave it open until midnight, I guess. If, yeah, I'm not entirely sure how many people we're missing right now, but I'll just leave it open until midnight and I'll send it in the Discord. So if you want to fill that out, or ask questions of each other, if not, you're free to go. And I'll share the results probably tomorrow.
Before I leave, actually, I think I forgot to mention that one of the new criteria for silver was demonstrating engineering success. So, like pretty much what that means is, I mean, showing design, build, test, but then also showing, I think, iteration. So once you get your results, you guys should also try to consider how will you improve those, like your new part that you just made or your product. But yeah, that's pretty. Yeah, that just reminded me. I mean, what Dr. Bartel said reminded me of that. But yeah, I mean, yeah, like they said, you guys did a really good job. So, yeah, it's pretty cool seeing you guys do all this research, and like what the professor said, you guys aren't expected to know everything, which is why advisors come in. Okay, on that note, I'm gonna leave. See you guys.
Okay, thank you for stopping by.
Yeah, no problem. Thanks for having me.